Here's a fact that the ND plus supplement industry would prefer you not think about too carefully. Every single ND plus precursor on the market, niacin, nicotinamide riboside, and NMN, produces the same downstream byproduct. That byproduct is nicotinamide, also known as NM. And nicotinamide, at elevated concentrations, directly inhibits the sirtuins that ND plus supplementation is supposed to activate. The entire enterprise of taking ND plus precursors to support sirtuin function contains within it the seeds of its own contradiction, and this is not a niche concern whispered in academic papers. It's a straightforward biochemical reality that should inform how anyone approaches long-term NAD plus supplementation. Sirtuins use NAD plus as a co-substrate to perform their deacetylase function, the molecular work of removing regulatory acetyl groups from target proteins involved in aging, metabolism, and DNA repair. When a sirtuin acts on its substrate, NAD plus is consumed and nicotinamide is released as a byproduct. Nicotinamide then acts as a feedback inhibitor, binding to the active site of sirtuins and slowing their activity. This is a regulatory mechanism. The cell uses nicotinamide accumulation as a signal that sirtuin activity is already high and should be moderated. It's homeostatic biology doing what homeostatic biology does. The problem emerges when supplementation artificially elevates ND plus turnover, generating more nicotinamide than the cell's clearance mechanisms can handle efficiently. What makes this particularly noteworthy is that NMN marketing has sometimes implied, without stating it explicitly, that its pathway avoids the nicotinamide accumulation problem that older compounds face. This implication is biochemically false. NMN is converted to NR to nicotinamide mononucleotide, but eventually, as ND+ is consumed in normal cellular reactions, nicotinamide is released. The endpoint of any NAD+ precursor supplementation strategy over time is nicotinamide accumulation. The pathway taken to get to NAD+ doesn't change what happens when ND+ is used. The sirtuin inhibition problem is shared equally by all precursors. The practical response to this involves two main strategies. The first is cycling, taking ND+ precursors for a defined period, then pausing to allow nicotinamide clearance before resuming. Protocols commonly suggested involve 2 to 4 weeks of supplementation followed by 1 week off, though optimal cycling parameters haven't been formally established in human trials. The second strategy involves supporting the methylation pathway that clears nicotinamide from the body. Nicotinamide is methylated and then excreted, and this process requires methyl groups donated by compounds like B12, folate, and trimethylglycine, also called TMG or betaine. Heavy ND+ supplementation without attention to methylation support may deplete methyl group reserves, a concern raised by some researchers, though the clinical magnitude of this effect in typical supplementation scenarios remains uncertain. For consumers investing significantly in NMN specifically because they believe it avoids problems associated with older compounds, this is a genuinely important correction. It doesn't necessarily mean ND+ supplementation is futile. The ND+ elevation itself may still produce benefits even alongside some nicotinamide accumulation. But it does mean that the sirtuin story is more complicated than most supplement marketing acknowledges, and that the assumed superiority of NMN's pathway over niacins in this specific domain is not scientifically supported.